studied microorganisms. In that one we have studied there is a fungus called the yeast. So as I said, yeast make the dough will rise, that is the wheat flour or maybe the maida. So during the preparation of bread, cake or etc. So now we are conducting how it will respire. As I said, sugar is required to grow for the yeast. So here it is one experiment we are conducting. So I am conducting one experiment. So it is easy to conduct, but don't conduct at home. So when you come to school, so we are doing together. So the 4 beakers I have taken. You have to take 4 beakers. This is according to your activity, how it will release carbon dioxide. So the first one beaker, it is a boiling water. So no sugar solution or no yeast crystals. So first I will show, this is the yeast crystals. So powdery like substance. So it is a synthetic one. Second, only lukewarm water and yeast. So I will shake it nicely. So then the third beaker, keep in your mind the first beaker just water, second beaker water with the yeast, third beaker sugar crystals with water, and the fourth one is very important. Now our result will come in that which one. So we have to identify. So in this beaker I have already taken water, yeast and sugar. So I will shake this jar and a beaker. So what we have to do now to know whether it is releasing carbon dioxide. So I will tie each jar or each beaker with a balloon. So the first beaker, the second with the blue color. We can take any color, but for the identification, keep in your mind the second is only the sugar solution, the third one yeast with water, and fourth one only boiling water. So keep for 6 hours at least or 8 hours. What is the results? So we can identify the respiration process, that is in the yeast cell, after few hours. So now we are conducting one experiment. So that is called using a yeast cell, how it will convert. So the glucose will be converted into a carbon dioxide, will be absorbed through this experiment, and the carbon dioxide will turn lime water in the form. So that we have done one interesting topic in this project. So the experiment is as follows. We have conducted already the first one, that is in first beaker A, it is just a boiling water. So only water. So I fixed a balloon. Next one, the B. The beaker B consists of water and yeast. Beaker B consists of water with the yeast fitted with a balloon. Beaker C consists of water and sugar, that is nothing but sugar solution. So this consists of sugar with water fixed with a balloon. The last one, so we are taking the beaker D consists of water, sugar plus yeast. So this beaker, so already we have conducted experiment. I kept for 3 hours, 3 to 4 hours. So what is the result in the out of 4? The first one only boiling water, second one water with the yeast, lukewarm water with the yeast, third one lukewarm water with the sugar, the fourth one sugar, yeast and lukewarm water I mixed and I fitted with a balloon. So now the result you can observe clearly. So here there is no rise of balloons in the 3, and the fourth one the balloon is raised. What is the inference? So if I take the balloon D, so the beaker D consists of why it is raised. The reason is when the yeast as a microorganism, it requires some media to respire. So that is sugar. So I have taken sugar. Sugar is the media consists of glucose. The glucose will be converted converted into an acid, and then these ones, especially the glucose, will be converted into an alcohol, and then it is respiring. So the raised in the structure of the balloon. So that is because of releasing of carbon dioxide. When the yeast will be mixed with a sugar solution in a lukewarm water, so it will not raise in cold water. The reason is requires certain temperature and certain media, that is sugar with a lukewarm water and the water. So yeast will raise in structure, releases carbon dioxide. So this is our inference. So don't conduct any experiment at home. So we are conducting here all the experiments. So just observe. So this is the one more microorganism is called the yeast, releasing of carbon dioxide. So now we are conducting one more using, so this, take the lid off, so remove the balloon slowly, holding your finger. So that is, so I am taking the carbon dioxide. What I do is, so this is a 2 test tube I am taking here. So I will fix this balloon, so to the lime water already I have taken in a test tube. What will is in your inference? So slowly you have to fill because it is already, it is a carbon dioxide. So I will fit this balloon to the test tube. So now observe carefully. So the water, lime water turns milky. So that indicates, you can see. So first it will be cleared water, now a white layer, a formation of white layer. The water converts, the lime water turns milky. The reason is, so carbon dioxide when it mixes with an lime water, so it turns milky. The presence, the indication there is a presence of carbon dioxide that is exhaling during respiration. So these are the experiment we have conducted. No need to conduct any experiment at home because require some precautions. So maybe the release of the carbon dioxide, maybe it is a danger at home. So while you are conducting an experiment, maybe the breaking of glass vessels or maybe the plastic container, etc. That's why, so my request is not to conduct any experiment at home, whichever it is easy, like before, that is a microorganism, you can prepare organic pit or a manure pit that you can conduct, but this experiment no need to conduct at home, we are conducting in our practical classes. So till now we have studied all the microorganisms and all the processes and how we have to preserve our food, like preservation of food. The last topic from your lesson, that is called nitrogen fixation. Nitrogen fixation. As we know, 78% nitrogen is present because in the food chain there is a high amount of plants. So that's why they are called the producers. They synthesize food. So highest number because they'll absorb sunlight. So during a process called photosynthesis, that is the preparing of food or synthesizing of food from their own. That is why they are called the autotrophs. So now, what is nitrogen fixation? And you have to study according to, maybe in your higher section you will study about the different types of biogeochemical cycle. Bio, living, geo, related to earth, a cycle, biogeochemical cycle. So which are the types? How many types of biogeochemical cycles are there in your higher section you will study? That is called nitrogen cycle, carbon cycle, water cycle, oxygen cycle. So but this year you are going to study only the nitrogen cycle in a systematic way. So nitrogen fixation, why it is made? For example, in the atmosphere, if you observe, so there is an atmospheric nitrogen. So this atmospheric nitrogen will not be utilized by plants as it is. So it requires converting into a nitrogenous compound, then only it is useful. So while studying this one nitrogen fixation, let us study the plants also with the root. So here just I have drawn the root. So identification of, so nitrogen fixation. in our mind, the question will arise that how the atmospheric nitrogen will fix and it will convert into nitrogenous compound. For example, in plants. So plants will convert, they will absorb in the root. So in the root they will absorb atmospheric nitrogen, and then on the root surface there is a structure called root nodules. Observe carefully, like knot-like structure in legume plant, like a pea plant. So these are the root in the dipot plant. The knot-like structure called the root nodule. These are nothing but rhizobium. Rhizobium. So this is the bacteria name. Rhizobium anabena nosta. So especially on the root surface, these knot-like structure. This is nothing but bacteria called rhizobium. So whether it will affect the root? No. So that's why they are called symbiotic. So if you are not harming another, but you are taking a shelter one another for the food, etc. So that is called symbiotically. So when the bacteria will present on the surface of the root nodule or the root, this is called nodule. So these will get the food from the root, and in turn bacteria will give back nitrogen to the root. After that this nitrogen will be utilized by the plant. So that is the nitrogen fixation biologically. So before studying there are the 4 points in nitrogen cycle. The first one, biological fixation, electrochemical fixation, artificial fixation and nitrogen fixation. So we will study now from the nitrogenous cycle. For example, as I said, atmospheric nitrogen is present in atmosphere. We have studied already. In atmosphere, oxygen, carbon dioxide, nitrogen and other compounds are present, other elements. So in that nitrogen, atmospheric nitrogen, imagine, so the total in the classroom or maybe in the outside atmosphere, atmospheric nitrogen is present. Whether the plant will take as it is? No. That's why, so during this atmospheric nitrogen, it will convert into nitrogen fixing bacteria. Nitrogen fixing bacteria, rhizobium. Nitrogen fixing bacteria fix atmospheric nitrogen. Think, imagine in atmosphere, nitrogen, atmospheric nitrogen is present. So which one will fix? The bacteria present. Nitrogen fixing bacteria. Nitrogen fixing bacteria fix atmospheric nitrogen. That is the first stage. What is the first stage? Atmospheric nitrogen will convert into, it will absorb by the bacteria. So that's why nitrogen fixing bacteria fix atmospheric nitrogen. Whether it is enough? No. One more step. It will convert into bacteria in turn. For example, atmospheric nitrogen will be fixed by nitrogen fixing bacteria. This bacteria in turn, so this bacterium in turn, fix nitrogen into nitrogenous compound. As I said, the plant require nitrogenous compound. So the root specially now will fix. Think here nitrogen will be there. So first nitrogen fixing bacteria will absorb atmospheric nitrogen, and then the bacteria present, rhizobium or other. So it will consist of a nitrogen, and then in turn nitrogen into the nitrogenous compound. Now it is on the root. So next one, after that it will convert compounds of nitrogen in the soil. So now in the soil, what is that present? It is present the nitrogen. Compounds of nitrogen in the soil. Next, so now it is present in the soil. This one will be absorbed. Bacteria turn compounds of nitrogen into bacteria is present, so it will convert the nitrogenous compound. So Nitrobacter, Pseudomonas, etc. Here the compounds of nitrogen in the soil will again convert into a gaseous nitrogen. So these gaseous nitrogen, for example, in the soil now nitrogen is present. Again the bacteria, a few bacteria will convert this nitrogenous compound into free nitrogen. That's why it is a gaseous nitrogen. These gaseous nitrogen again released to the atmosphere. So now again it is atmospheric nitrogen. I repeat the step. Atmospheric nitrogen in the atmosphere, it will be absorbed by nitrogen fixing bacteria, fix atmospheric nitrogen, then it will convert into a bacteria. In turn, the bacteria will in turn convert into a nitrogenous compound, and now it is present in the soil. Again, so few bacteria will convert this into a gaseous form, and again it will be released to the atmosphere. So this is the process. For example, now I will explain once again. Think here atmospheric nitrogen. So this will be absorbed by the bacteria, and it will fix nitrogenous compound, and this nitrogenous compound will enter into the soil. And in the soil, again the bacteria will convert into a gaseous form, and it will enter atmosphere. That is the first step. That is called biological fixation. Biologically means using living organism like Rhizobium, Anabaena, nostoc. If you observe on the paddy field or in the pond water, so we can observe Rhizobium, Anabaena, nostoc, etc. So some are algae, some are bacteria. So mainly now the bacteria Rhizobium. The next one, electrochemical fixation. Electric chemical fixation. So during lightning, these atmospheric nitrogen. So during lightning, these rays, the electrical ray, then it will combine with oxygen present in the nature. In turn, it will convert into oxides of nitrogen. Oxides of nitrogen, which will combine with rain, and then it turn into, so it will fall to the earth's surface as a nitric acid with the water. So rainwater consists of a nitric acid, and when it will fix to the soil, so these when combines with oxygen, it forms an oxides of nitrogen. These oxides of nitrogen will fix to the soil, so in the form of nitric acid, and it will fall to the earth's surface as rain, and then in that compounds of nitrogen in the soil. So that's why they are called electrochemical. Electro means lightning. It consists of an high electrical power. Electrochemical. It converts into combines with oxygen, forms oxides of nitrogen. So that's why it is called chemical. And in turn it will convert into a nitric acid, dilute nitric acid in the form of rain, and then it will fall to the earth's surface. So that's why compounds of nitrogen in the soil, that is called electrochemical fixation. After that, for example, compounds of nitrogen in the soil, one we have studied, the bacteria will convert into again gaseous form. Another. So whatever it is present in the soil, nitrogen will be absorbed by the plants, will be eaten by the uptake by the plants. The plants, especially these plants from the soil, absorb nitrogen. The plants will absorb the nitrogen from the soil. That is the meaning uptake by plants. Now these plants will be eaten by animals. Any animals will get protein from plants. After that, there is an excretion process. So nitrogenous waste from excretion, when it will release, again it will leads to the soil. So that's why again all the three compounds. First, compounds of nitrogen in the soil absorbed by the plants. The plants will be eaten by animals. After excretion, these excretion is nothing but ammonia. Will convert into a nitrogenous compound. So that's why there are the three processes: ammonification. So that's the plant will absorb the protein and then it will release as an excretory substance ammonia. Ammonia will convert into a nitrogenous compound that is called nitrogen fixation or nitrification, and again the nitrification into a denitrification that is called the gaseous nitrogen. So this is related to your nitrogen cycle. Artificial fixation, as it is present in the soil, it will be fixed in the soil itself, nitrogen. Because of some reason, the oxygen will combine with the nitrogen, it will form a different component that is called artificial fixation. So nitrogen fixation again by bacteria like rhizobium and maybe few bacteria like Pseudomonas and Anabaena, nostoc, etc. So all these are related to our nitrogen cycle. Why it is required? It is required to survive the plant, mainly in a food chain. The first layer, the first step that is called the producer, they prepare their own food using sunlight. That's why that process is called photosynthesis. By that, the next group of animals will get the food. So that's why the plant is necessary on the earth surface. These plants will absorb sunlight. They prepare their own food. So the rhizobium will help to give the nitrogen. So that's why it is called nitrogen fixation. Again I repeat, atmospheric nitrogen converts into a nitrogen fixing bacteria, fix atmospheric nitrogen. Again it will turn into bacteria and turn fixed nitrogen into nitrogenous compound, and then again it will turn into compounds of nitrogen in the soil. Again the bacteria will convert, in turn converts the nitrogen into gaseous form. This gaseous nitrogen will release to the atmospheric nitrogen. This cycle will be, so why it is in the form of a spherical cycle? The reason is it will be continuous till the life will be existed. So that's why there is a one, and from the lightning you will get electrochemical fixation to the soil. That is the nitrogen, and the plants will absorb the nitrogen from the soil. This will be eaten by animals, and from the excretory ammonia will convert into a nitrogenous compound. Again the waste excretion released to the soil. So by that it will convert again into a gaseous nitrogen. It is in a complete cycle. So we have studied now nitrogen fixation, and let us conduct few activities through an diagram and through an experiments. So now I will explain the activity 2.5. Activity 2.5. So that is related how the microorganism converts a waste material into an manure. So that is how useful it is, a microorganism, so which is beneficial to us. So that is a one in activity 2.5. So take a 2 pot, label it A. The first one is A, the second one is B. So now what we have to do? In the first pot, fill the soil. So till half or more than half. What you have to fill? You have to fill soil. What about the pot B? Again, here also half of the pot will be filled with soil. Next. So in the first pot, pot A, you have to fill the waste material like leaf of the cabbage or maybe onion leaves. All the waste material, all the waste material means organic waste. For example, leaves or maybe vegetable waste or etc. So this will be in the pot A. Pot A consists of waste material like leaves, vegetable peels, etc. So as waste material, after if you are using So some of the vegetable and the fruit, leaves, etc. So waste material, whatever it is there, you have to fill in the pot A. Now, so whatever the plastic containers you are using, so that you have to fill in the pot B. So here plastic materials and bottles, etc. So you have to fill here, and then maybe the used pens. So that is also you can use it. So the first one, soil with the waste leaves, maybe vegetable peels, etc. Next, pot B consists of soil and add plastic materials to it. Keep for several days. What is your observation? So we have to find out. So the result. Now in pot A, what is the result we can see? The pot A, the soil will mix with all the vegetable waste or the leaves, etc. It convert into a component called manure. So why it is useful? So when I mix with, so maybe now at present recently we are giving an opportunity to make our own organic manure. That is so to give the food to our plants, to our garden, or maybe for the vegetable plants, we are preparing the manure in kitchen garden. So that is the one. So now this will be converted into manure. There is a question mark. How it is converted into a manure? Everything will be decomposed. Decomposed by dash. It is decomposed by bacteria. So that's why bacteria is a microorganism. It is beneficial to us to convert the dead and decayed plants. Dead plants and animals will be eaten by bacteria, convert into manure. This manure nothing but inorganic soil or inorganic compounds. These inorganic compounds will be used by dash. It will be used by plants. So it is used by the plants. So whatever the inorganic material compound, the term will say the manure, it will be absorbed by the root. The plant will grow healthy because it is a good manure. So it is because of a component that decomposes bacteria, fungi, etc. So fungi are also called as saprophytes. They feed on dead and decay bacteria, fungi, etc. So they convert. Somehow the earthworm will convert only a few material, but bacteria will convert the entire waste material, that is the dead plants and animals. They decompose into a component manure. That manure it is called an inorganic compound, but we cannot use the inorganic compound. So this as intaking. So if we intake plants, so we will get organic compound. They prepare the food, so that is in the form of organic compound using inorganic soil. So that is in the first. It's very good. There is an activity given how microorganism is beneficial and how it will control. In the second. So what we have taken? Soil, plastic materials, polythenes, etc. cover everything. So whether it is decomposed? No. The reason is all these materials in the soil, maybe bacteria is present, but the plastic materials will be not eaten by the bacteria. The reason is they are non-biodegradable. Non-biodegradable. The things which cannot be decomposed by microorganisms, that is called non-biodegradable. So that's why, so not to put together in our kitchen. So the reason is, so if I pour or if I throw all the plastic material, So in between with the decayed substance, whether it will be decomposed, no. So then it will give the pungent smell also, and especially here the plastic material will block the root of a plant. So there will be hidden growth of the plants, cannot, and then this will not be decayed. So that's why that is called the non-biodegradable. It will remain as it is. In that, so the observation is, so we need bacteria to convert the plant materials, so the dead end plants and animals. So we require microorganism. That's why we can prove that microorganism is an beneficial one and it is very useful to us. That is one activity. The second activity is given according to the yeast cell as a microorganism. Already we have conducted experiment now. So then, so after 3 or 6 hours, especially after 6 hours, what will happen? So whether any fermentation will take place, what is the result? Let us see. So now I'll explain the result, then I'll show the observation of that experiment.